Good morning, good day. So we have here the illustration, the African skin colors, the different gamemelas, and we have the pea plants. Our topic is all about non-Mendelian inheritance. So this is called non-Mendelian inheritance and it plays an important role in several disease processes. Non-Mendelian inheritance include incomplete dominance, co-dominance, multiple alleles, pleiotrophy, lethality, polygenic traits, and we have environmental factors. First is incomplete dominance. It states that it is when a dominant allele or form of a gene does not completely mask the effects of a recessive allele, and the organism's resulting physical appearance shows a blending of both alleles. Blending is the key word for incomplete dominance. Incomplete dominance is an important concept in the study of genetics. It refers to a circumstance in which the two copies of a gene for a particular trait or alleles combine so that neither dominates the other. No one dominates the other. This creates a new phenotype or set of observable characteristics caused by the interaction of genetics and environment. In short, incomplete dominance is when neither gene is fully dominant and the result is a brand new trait. Non-Mendelian inheritance can manifest as incomplete dominance, where offspring do not display traits of either parent but rather a mix of both. Two alleles produce an intermediate phenotype rather than either one exerting a specific dominance. Incomplete dominance will give a 1:2:1 phenotypic ratio with the homozygous genotypes each showing a different feature and the heterozygous showing one more distinct phenotype. Incomplete dominance is also called semi-dominance or partial dominance. One example is shown in rose. So we have rose. The allele for red color is dominant over the allele for white color, but heterozygous roses, which have both alleles, are pink. So note that this is different from codominance, which is when both alleles are expressed at the same time. So meaning in incomplete dominance, neither of the parents' allele are seen. Okay, so incomplete dominance. It is important to note that most observable traits in any living thing are caused by more than one gene. Incomplete dominance is specific to traits that occur on just one gene. However, there are many such traits, and incomplete dominance occurs in every sort of organisms that has genes, including plants, animals, and even human beings. So why does incomplete dominance occur? As we have seen, it does not always occur with flower color. Roses and tulips, carnations, and snapdragons, among others, show incomplete dominance, but Mendel's pea plants showed complete dominance. Incomplete dominance can occur because neither of the two alleles is fully dominant over the other, or because the dominant allele does not fully dominate the recessive allele. So this results in a phenotype that is different from both the dominant and recessive alleles, and appears to be a mixture of both. Example of an incomplete dominance in humans. So a child born to a parent with straight hair and a parent with a curly hair will usually have wavy hair, or hair that is a little curled, due to the expression of both curly and straight alleles. So incomplete dominance can be seen in many other physical characteristics such as skin color, height, hand size, and vocal pitch. So in other animals, we have the Andalusian chicken. So it is a type of chicken native to the Andalusia region of Spain. Shows incomplete dominance in its feather color. So we have here a white male and a black female will often produce offspring that have blue tinged feathers. So this is caused by a dilution gene that partially dilutes the pigment melanin and makes the feather lighter. So this is incomplete dominance. So let's test. Which is not an example of incomplete dominance? A. Pink flower produced from red and white flowers. B. A flower that is both red and white produced from red and white flowers. C. Curly-haired and straight-haired individuals producing wavy hair. D. A highly spotted dog and a non-spotted dog producing puppies with a few spots. Go. Okay, what is your answer? Okay, the correct answer is letter B. A flower that is both red and white produced from red and white flower. So letter B is correct. So this is an example of codominance, not incomplete dominance, because both phenotypes are shown instead of one intermediate phenotype. So we have codominance. Co meaning complementary dominance. It states that it occurs when two versions or alleles of the same gene are present in a living team and both are present. So the key word here is both are expressed. So non-Mendelian inheritance is applicable in codominance, where two alleles may be expressed simultaneously, that is heterozygous. There is no mixing or blending involved. The human blood type AB, where types A and B are both codominant, is an example of this. A cross between AA and homozygous BB will produce AB offspring, with both alleles being expressed equally. Often, codominance is linked with a characteristic that has multiple alleles of a given gene. In many cases, one of those alleles will be recessive and two others will be dominant. So this gives a trait the ability to follow the Mendelian laws of heredity with simple or complete dominance, or alternatively, to have a situation where codominance occurs. For example, coat color of rabbits can appear as four common phenotypes. So black or full color brown rabbit, and we have chinchilla, the Himalayan, and albino. So we have full color brown is dominant over all the others. Chinchilla is dominant over only two Himalayan and albino, and Himalayan is dominant over albino, and albino is recessive to recessive to all. So in this case, the full color or brown is what? Is completely dominant all the others. So the chinchilla is incompletely dominant to the Himalayan and albino alleles. We have the Himalayan allele is completely dominant to the albino allele, and the albino rabbit is recessive to all rabbits. So instead of one trait being dominant over the other, both traits appear. So codominance is easy to spot in plants and animals that have more than one pigment color. For example, spotted cows, which is a combination of black and white caps. And we have flowers with petals of two different colors are example of co-dominance. So next is we have example of co-dominance in livestock. When a chicken with white feathers breeds with a chicken with black feathers, so what is the result? So the result is an offspring chicken that grows up to have both the black and white chicken. So likewise, when a red cattle breeds with a white cattle, the resulting offspring may what? May show both red and white hairs, resulting a mixed coat pattern called roan. So we have here redundance, and other flowers may also exhibit codominance. So the crossing of a red chamomilla and a white chamomilla may yield a flower that has what? That has both red and white patches. So many flowers show similar patterns of codominance where both of the parental flower colors show up different parts of the plant. So in blood type, an example of codominance that occurs in human is that of the blood type. So there are three different versions of the gene, four proteins that appear on the outside of our blood cells and help our body to identify the cells as their own. So these alleles are A, B, and O. So the O allele actually does not code for any protein at all. So people with the O trait lack both A and B proteins. The A and B proteins, on the other hand, code for two different proteins. So these proteins, like different colors in a flower, can appear together. Someone who inherits an A allele from one parent and B allele from the other will express what? Both parents in a codominant fashion, resulting in an A-B blood type. So the O trait, on the other hand, is a good example of a dominant or recessive relationship if either A or B is expressed. The O trait is not expressed. So this chart illustrates how codominance can occur between A and B traits, while a dominant or recessive relationship exists between those traits and the O trait. Let's check your understanding. Which of the following is not an example of codominance? A. A child of parents with blood types A and B who has AB blood type. B. A calf of a red cow and a white cow who has roan coat consisting of red and white hairs. C. A child of a parent with blue eyes and a parent with brown eyes who has brown eyes. D. A flower of offspring of red and white flowers which has both red and white petals. Go. Okay, what is your answer? The correct answer is letter C. A child of a parent with blue eyes and a parent with brown eyes. Letter C is correct. So this is not an example of codominance because the child does not express both parents' traits. She only expresses the brown-eyed gene inherited from one parent. So next is multiple alleles. Multiple alleles exist in a population when there are many variations of a gene present. So in organism with two copies of every gene, also known as diploid organisms, each organism has the ability to express two alleles at the same time. So keyword is a variation of a gene. So multiple alleles exist in a population when there are many variations of a gene present. So they can be the same allele, which is called homozygous genotype, Alternatively, the genotype can consist of alleles of different types known as heterozygous genotype. So haploid organisms and cells only have one copy of a gene, but the population can still have many alleles even though they have only one copy of a gene. So multiple alleles combine in different ways in a population and produce different phenotypes. So these phenotypes are caused by the proteins encoded for by the various alleles. Although each gene encodes for the same type of protein, the different alleles can cause high variability in the functioning of these proteins. Just because a protein functions at a higher or lower rate does not make it good or bad. So this is determined by the sum of the interactions of all proteins produced in an organism and the effects of the environment of those proteins. So are some organisms rather driven by multiple alleles in a variety of genes, do better than others, and can reproduce more. So this is the basis of natural selection, and new mutations arise and new lines of genetics are born. The origin of species takes place. So example of multiple alleles, so coat colors in cats. So in domestic cats, breeding has taken place for thousands of years, selecting for different and varied coat colors. Cats can be seen with long hair, short hair, or with no hair. There are genes that code for whether or not a cat will have hair. So there are multiple alleles for this gene, some that produce hairless cats, and some that produce cats with hair. Another gene regulates the length of the hair. Long-haired cats have two recessive alleles, while a dominant allele will produce a what? A short hair. So other genes control the color of coat. So there is a gene for several colors of pigment: red, black, and brown. Each gene has multiple alleles in the population, which expresses the protein responsible for making the pigment. So each allele changes the way the protein works, and therefore what? The expression of the pigment in the cat. Other genes, in similar ways, control traits for curliness, shading, patterns, and even textures. So the amount of combination and expression of different genotypes together creates an almost infinite variety of cats. For this reason, cat breeders have been successfully attempting for thousands of years to create new and strange varieties of cats and dogs for that matter, even with only four alleles between two parents at each gene. The variety can be incredible. Just look at the kittens in the photo. All these kittens came from the same parents. Another example of multiple allele is we have the fruit flies. In the year 2000 or 2000, scientists finally succeeded in mapping the complex genome of the common fruit fly known as we have the Drosophila melanogaster. So the fruit fly had been and continues to be a valuable laboratory animal because of its high reproduction rate and the simplicity of keeping and analyzing large quantities of flies at about 165 million base pairs. The DNA of a fruit fly is much smaller than that of a human. While a human has only 23 chromosomes pair, or 46 chromosomes a total, so a fruit fly only has 4 chromosomes. Still, in only 4 chromosomes, there exist around 17,000 genes. Each gene controls a different speck of a fly and is subject to mutation and new alleles arising. So in the picture, all the flies are the same species, known as Drosophila melanogaster. The variation seen between the flies is caused by multiple alleles in different genes. For instance, the gene for eye color determines if the fly will have an orange or a brown eye, or even a white eye. Amazing. So both the white and orange alleles are recessive to the wild type red eye allele. So the two flies at the top have wild type bodies, a tan and dark stripes. In the gene that controls body color, two other colors are present. So the fly on the far right is shown a homozygous recessive genotype that causes a dark body. So the three flies on the bottom show another homozygous recessive genotype, the yellow body mutation. So this is an example of multiple alleles. Let's check your understanding. Often, breeders of animals aim to breed through lines. This means that generation after generation, the animals will look almost exactly the same, and the number of different alleles in a population is reduced. Why would this be important for scientific research? A. It is not important. B. Stable organisms ensure that the experiment can be repeated. Or letter C. More variety is good for research. Go. Okay, what is your answer? The correct answer is the letter B. Stable organisms ensure that the experiment can be repeated. Letter B is correct. Why? So in a research environment, you want as little variation as possible. This makes the result more meaningful. If lines are bred through, then organisms can be bred for generations and produce the same results as when the experiments were started. Without this decrease in variation through artificial selection, many experiments would not be reproducible. Being able to reproduce an experiment further is the basis of all good science. Next, we have the pleiotrophy. Pleiotrophy. So pleiotrophy, it refers to the expression of multiple traits by a single gene. So these expressed traits may or may not be related. Pleiotrophy was first noticed by geneticists. We have Gregor Mendel, who is known for his famous studies with pea plant, Pisum sativum. Mendel noticed that plant flower color, white or purple, has always related to the color of the leaf axil, or the area on the plant stem consisting of the angle between the leaf rudder and upper part of the stem and the seed coat. So the study of pleiotrophic genes is important to genetics, as it helps us to understand how certain traits are linked in genetic diseases. Pleiotropy can be spoken of in various forms. We have several forms of pleiotropy. We have the gene pleiotropy, the developmental, we have the selectional, and we have the antagonistic. So pleiotropy is the expression of multiple traits by a single gene. Keyword for pleiotropy is multiple traits by a single gene. Multiple traits by a single gene. Okay, we have gene pleiotropy. Is focused on the number of traits and biochemical factors impacted by a gene. So, number of traits and biochemical factors impacted by a gene. And we have developmental. So it is focused in what? On mutations and their influence on multiple traits development. Selectional. is focused on the number of separate fitness components affected by a gene mutation, and we have the antagonist is focused on the prevalence of gene mutations that have advantages early in life and disadvantages late in life. Okay, so what is an example of pleiotrophy? So an example of this that occurs in human is sickle cell disease. So this one. So when we say sickle cell disorder results from the development of abnormally shaped red blood cells. Normal red blood cells have a biconcave disc-like shape and contain innermost amounts of a protein called hemoglobin. So hemoglobin helps red blood cells bind to and transport oxygen to cells and tissues of the body. So sickle cell is a result of what? Of a mutation in a beta globin gene. This mutation results in red blood cells that are sickle-shaped, which causes them to clump together and become stuck in blood vessels, blocking our normal blood flow. So the single mutation of the beta globin gene results in various health complications and causes damage to multiple organs, including the heart, brain, and we have the lungs. Another example of this is the PKU or the phenylketonuria, another disease resulting from pleiotrophy. So PKU is caused by a mutation of the gene responsible for the production of an enzyme called phenylalanine hydroxylase. And another example is the frizzled feather trait. Next, combinations of genes are often needed to promote the survival of organisms. If an allele that contributes to a gene is not expressed, it can lead to harmful activity. This is known as lethality. So inheriting two copies of mutated genes that are non-functional can have little consequences. So a classic example of an allele that affects survival is that lethal yellow allele, a spontaneous mutation in mice that makes their coats colored yellow. So mice that are homozygous die early in the development. Although this particular allele is dominant, lethal alleles can be dominant or recessive and can be expressed in homozygous or heterozygous condition. So we have an example of lethality. We have white mice. So some characteristics are polygenic, meaning that they are controlled by a number of different or multiple genes. So in polygenic inheritance, traits often form a phenotypic spectrum rather than falling into clear-cut categories. So phylogenetic traits are traits that are controlled by multiple genes instead of just one, controlled by multiple genes instead of one. So the genes that control them may be located near each other or even in separate chromosomes. Because multiple genes are involved, polygenic traits do not follow Mendel's pattern of inheritance. Instead of being measured discretely, they are often represented as a range of continuous variation. Some examples of polygenic traits are the height, your skin color, the eye color, and we have the hair color. So these are examples of polygenic traits. Height. So the human height is controlled by many genes. In fact, there are over 400 genes related to height, and all of these genes interact to make up a person's phenotype. This is a very large number, but it makes sense because height is a compilation of the lengths of many different body parts such as leg bones, the torso, and even our neck. So polygenic traits can also be influenced by an organism's environmental factors. So if a person gets inadequate nutrition during childhood, they can have stunted growth and end up smaller and shorter than they would otherwise. So it is estimated that 90% of a person's adult height is controlled by our genetics, and 10% is affected by the environment. Next example of polygenic trait is the skin color. Skin color. So in human, skin color is influenced by many things, but the pigment of melanin water influences most of a person's phenotype. In general, the more melanin a person has, the darker their skin is. So albino, for example, people produce no melanin at all. So the body creates more melanin to protect against the sun's ultraviolet rays, which is why skin darkens after prolonged sun exposure. The amount and type of melanin that a person produces, such as eomelanin, pheomelanin, and euromelanin, is controlled by multiple genes rather, and the different types of melanin interact to form the final phenotype. For example, people with red hair have more pheomelanin and often have a pinkish skin tone. Okay, next. We have the next example of phylogenetic trait, which is our eye color. Our eye color. So there are two major human eye color genes, the OCA2, the HERC2. But at least 13 other genes also play a role. So the colored part of a person's eye is the iris. It is a muscle that changes the size of the pupil in order to change the amount of light that is absorbed by the retina. A person's eye color is determined by the pigmentation of their irises, but also by the way the cells in their irises scatter light. As with skin color, eye color is affected by the presence of melanin. People with brown eyes, like me, have a lot of melanin, while people with blue eyes have low melanin in the front part of the iris that is visible. So green eyes are caused by multiple factors. They are the result of a light brown iris combined with a blue tone given by light scattering. So that is an example of the phylogenetic traits. Let's check your understanding. Which of these is not a polygenic trait? A. Height, B. Skin color, C. Eye color, or D. Widow's peak? Go. Okay, what is your answer? The correct answer is letter D. Widow's peak is not a polygenic trait. So D is the answer. Why? So having a widow's peak, which is a V-shaped front of the hairline. So this one, I have a widow's peak. So V-shaped front of the hairline is not a polygenic trait. Individuals that have at least one dominant allele have a widow's peak, while individuals with two recessive alleles have a hairline that is straight across the forehead. So widow's peak is not a polygenic trait. So that would be all. Thank you for viewing and hoping meron kayong natutunan for this topic. Thank you.